Histology is the study of human tissues. So a tissue is a whole bunch of cells of the same type working together for some greater purpose. So we're going to be going up that hierarchy of organization from cellular level to the tissue level. And then this is our last stop before we move on to some actual organ systems. So these cells are all going to come from the same embryological region, and they perform a specific function. There are 4 types of tissues in the human body. Epithelial tissue. Epithelial tissue is typically found as a lining. So anytime the outside of something kind of meets the inside of something. So think about your skin. The skin is the lining between the outside of your body and the inside of your body. Same for your digestive tract, right? Your digestive tract is basically a tube of air all the way through your body from your mouth to the anus. There is a lining around that tube between the inside of the digestive tract and the outside of it, which is your body. Connective tissue tends to do kind of what it sounds like. It connects things together. It holds things together. Connective tissue is going to be things like bones and ligaments that are going to be supportive. We have nervous tissue, and nervous tissue does one thing, which is communication. And then we have muscular tissue, and muscular tissue is going to be involved in locomotion. So when you were an embryo, the very first few hours that you existed, you started out as a single cell, and that cell divided a bunch of times to make this little ball of cells that you see at the bottom, which is called a blastula, and you will talk about these more in anatomy too. But the very first thing you did to specialize your different cells was they started to turn into 3 different regions of cells. You have ectoderm, which if you look at these words, ecto, that means outside. So ectoderm is the cell layers that give rise to kind of the most superficial things. So that would be the skin and also your central nervous system, so your brain and spinal cord. Meso, so meso is middle. So this is kind of the middle layer, not the innermost layer, not the outermost layer. This is going to be things like your bones, muscles, cardiac muscle, heart, blood, some of your major organs. And then your inner layer, endo, means inside. So that's the innermost layer. So that's at the very center of you. That's that digestive tube that goes through your body. Your respiratory system, reproductive systems. Most of the endoderm is all of the stuff that has an outlet to some portion of your body, right? Your digestive system has an inlet at your mouth and an outlet at the anus. The respiratory system connects to your mouth. The reproductive system connects to the urethra, which exits the body. When we look at tissues, what we're going to be looking at is a lot of pictures of microscope slides. And you're probably going to see these kind of tissue sections in lab as well. So when a microscope slide is labeled, they'll be labeled as longitudinal sections, cross sections, oblique sections. And so this is what you're going to see. When you cut something down its long axis like this, that's called longitudinal. So what we've done here is, this is kind of a typical long bone, and then you've got just a tube. So this would be kind of like maybe a little small vessel. If we cut it down long ways, what you would see is just the 2 edges. We don't do this often for vessels, but you may see longitudinal sections for longer bones. A cross section is the opposite of that. So we're going to cut it horizontally through the middle. A cross-section is really common for studying small tubes. And then anything between that, so anything at any angle that's not perfectly vertical, up and down, or perfectly horizontal, left and right, is going to be called oblique, because it can be any angle. And that gives us a nice big area to look at under a microscope. So we do these kind of sections for things that are solid, like bone. We would definitely do a cross-section of bone. But for a liquid, so something like down here, we have blood. Liquid tissues are often prepared as smears. So what you'll do is you take a sample of the blood, put it on a microscope plate, and then put a cover slip over it and then actually seal it to preserve it. So take a moment and reflect on the 3 germ layers and what tissues are derived from each.